Hi everyone, my name is Amir Manzoor and I teach upstate New York at an institute called SUNY Polytechnic Institute. I'm very glad to have the opportunity to present our work at the conference, CIBCB conference. Unfortunately, we could not be there in person, but if there's anything that interests you, please contact us. This work is about using geometric deep learning to design TPP riboswitches. As we know, in the more recent years, using RNA devices for therapeutic purposes has been gaining a lot of attention, and people believe that RNA has the potential to tackle different diseases and cancers due to its great structural versatility and malleability. Indeed, there are many RNAs that are used as drugs or in therapeutics as a whole, such as siRNAs, antisense, oligonucleotides, and many more. So designing RNAs experimentally can be very expensive. Therefore, the state-of-the-art deep learning methods are currently being used to design sequences that fold into desired structures that can be used in therapeutics or design therapies. These methods usually are based on training their models on known structures, so they are exposed to known atomic resolution structures taken from different experiments. They're available publicly in different websites, PDB and RNA Solo, just to name a few. And the way they are assessed, their performance is assessed. They're assessed by many ways, but one classic way to, as a metric, would be to see how much the predicted sequence they have matches the template sequence. So if I were to explain this, once we train our model and we expose it to thousands of atomic resolution data, then during the test set, we expose it to a new structure that is never seen, and we mask the actual sequence. We ask the model to predict the sequence, and then we compare that prediction with the ground truth. And your sequence recovery would just be the percentage of nucleotides that are correctly predicted in their correct positions. And some of the more successful models here are R-design and ribodiffusion. These are they use transformers or diffusion models, as well as GNN, which is a multi-state RNA model, RNA design deep learning model using graph neural networks. This is very successful. And recently, we developed our own method based on the Shaitana Joshi's work. Based on GNN, we developed our own method that also uses graph neural networks, and we achieved the competitive results. I'll explain our method in a few slides and then talk about deploying it for the TPP riboswitch. So what we do is that we take the RNA structure and we identify the base pairs using an external software. We identify different types of chemical bonds, such as base pairs, backbone bonds, and other spatial kind of positioning of nucleotides. We use X3DNA or FRED or other software. Then the orientation of each nucleotide is determined from the PDB files. As you can see, each nucleotide has around 21 atoms, and their XYZ coordinates are available in the PDB file. We take three major ones. Generally, people take these atoms N1P and C4 prime. So we take those, and then that will help us determine an orientation for that nucleotide in 3D space. And then if I take that information for two nucleotides, for example, having a chemical bond, I basically have everything I need in terms of the distance between nucleotides and the orientation relative to the first nucleotide, for example. So all of that information here is basically used to convert the PDB file that represents a structure taken from experiments, into a graph, a mathematical structure that assigns nodes to each nucleotide and edges to each bond. So we also tag our edges, which are actually resembling bonds. We tag them with the type of the bond. For example, we have primary structure edges and secondary structure edges. We also use another component in our model, which is basically was developed with respect to other fields such as proteins, but it was very useful, which is called geometric vector perceptrons. What they do is that they kind of digest all of these scalar as well as the vector information for each, I would say, graph edge, and they kind of process that to memorize these orientations and these geometries. So this is the kind of the schematic of our own pipeline, which consists of multiple layers. We usually use two to four layers. That's sufficient. We expose the model with the structure, basically the graph. That is a graph that has geometry. Every node has a geometry, and the adjacency metrics of the edges are tagged by the type of that edge. We might have multiple structures that are available for a specific sequence, so we simultaneously feed that into the model if we had, let's say, 5 structures for the same sequence. And then we would combine that information using the GVP blocks. Basically, these are all GVP blocks, so we combine them based on different, each applied on their own edge type, and then we combine those information and the hidden embeddings to predict a sequence that basically could explain the structure. So during a test, the test said that sequence is predicted while the nucleotides are masked. So the model has no idea what the nucleotides are, but it predicts the nucleotides based on the geometry that it sees. We use message passing in our training, and we basically have different multiple layers to learn more about the neighbors of neighbors in the graph. So we would have around 2 layers here. Okay. So we achieved an accuracy of around 70% in this project. That was, we've done previously, which is fairly acceptable. It's actually competitive, and but this is more average across all RNAs. But we were more interested in how this model performs in specifically predicting sequences of TPP riboswitches, because if we could predict them properly or with a high accuracy, we would effectively have designed sequences that are hypothesized to fold into what the TPP riboswitch would fold into. So that's basically the whole idea of sequence design. We predict the sequences that fold into the same structure. So why TPP riboswitch? Well, TPP riboswitch is the only riboswitch that has been seen in eukaryotes as well. It performs gene expression by being exposed to the thymine pyrophosphate ligand, which is an active form of vitamin B1. And this is the 3D crystal structure that is resolved. So when the TPP is not available, RNA has another structure and it performs a different task. TPP riboswitch also serves as an antimicrobial drug target and is useful for medical applications, all of which are reasons why we focused on the TPP riboswitch. It's also used as a classic riboswitch historically. So we gathered around 27 riboswitches, most of them experimentally resolved in the bound state. We wanted to evaluate a few things. First, we wanted to see how successful our model is specifically on predicting the sequence of the TPP riboswitch. Then we wanted to know what should our training data sets have? What are the characteristics of a good training data set that we should feed our model? So then, in order to do that, we gathered 3 different data sets. One was just solo RNAs, which is around 1,000 sequences, as you can see here, mostly consisting of microRNAs and ribozymes. and other small RNAs, as well as long RNAs such as ribosomes. Then we have ribose switch data set, which is all the other ribose switches except TPP ribose switch, as you can see here. We also had a bigger data set available to us, around 15,000, much more than the other ones, which was RNA protein complexes. So we also extracted the RNA geometrical structures from these RNA protein complexes and used and put that in a different training set. So we had 3 different training sets. We also had 27 TPP ribose switches, and of course we needed a benchmark, so we used another kind of unique structure as a secondary test set. We use basically G quadruplexes because they have such a specific structure that's not similar to base pairing. So we wanted to see how our model behaves and how it performs when it is exposed to a structure it's never seen. So G4 was a good kind of a benchmark. And then we would compare the performance of predicting sequences of G4s versus predicting sequences of the TPP ribose switch, because none of the models are exposed to TPP or G4 structures. So first, we found that all models are successful in predicting sequences of structures that belong to the same set. For example, if I were to explain this graph and this table, for the solo RNA, we would initially takes, let's say, 10% of the sequences out as a test set. And then after we train our model on the 90%, we would apply the model parameters on that 10%. And what you see is the sequence recovery, for example, here is the performance. As you can see, the ribose switch has a good performance, around 75%, which is very good, higher than the average of what we would expect. But then we would take these models, and our task was investigating prediction of structures it's never seen, or structures that are not similar to what it has, which would be the TPP and G4. And to our surprise, the ribose switch data set did not do very well to predict sequences of the TPP ribose switch, even though TPP is also a ribose switch is just another ribose switch. But because of the fact that this ribose switch was not included in the training set, the performance was poor. And interestingly, the data set of the solo RNA had a higher performance, which was basically simple structures. And all of them had better performance than predicting G4. So predicting G4s was basically not very high. It was kind of expected because the structure is very unique, and there are other reasons in formation of G4s, which makes it very hard to predict its structure. And but what is striking is the performance of the solo RNAs, which was higher. So we kind of took a closer look in how our model predicts the bound state of the TPP versus the unbound state. We took 2 candidate predicted sequences, PRED1 and PRED2, which were derived from their corresponding bound state and non-bound state templates, which were experimentally resolved. And they had a very high sequence recovery. So these sequences are very similar to the original template. And we wanted to see if the structure is also very similar to the original template. So the RMSDs were not as low. Basically, RMSD is the structural comparison. And to compare the structures, we used AlphaFold because the structure of our prediction was not available. So we computationally predicted it. And as we had expected, the predicted sequences resembled the unbound state structure very, very well. Even though the angstrom are not very low, the L-shaped structure of the TPP ribose, which is very famous, is also formed by the green, which is our predicted sequence, our hypothesized sequence. So our AlphaFold predicted structure actually similar to the unbound state. I cannot say the same with the bound state. Our bound state structure prediction, which also used AlphaFold, is very different on the far right, very different than the original structure of the TPP. We even used TPP ligand in our AlphaFold prediction to create some sort of a more realistic scenario. But it looks like the sequences that we predict fold reasonably well when they're predicting unbound states, but when it comes to the bound states, they do not. I also would like to add here that, of course, some of this lack of good prediction in the bound state can be kind of imagined by other factors that are involved here, such as metal ions or the ligands kind of distorting the structure. But the length of the sequence is not an issue here. The good thing about, but I forgot to mention, the good thing about graph neural networks is that it is ambivalent to the length of the input. So it doesn't care how long the sequence is. You could train it on smaller structure, but apply to a larger structure. It kind of repeats the same thing for a larger number of nodes. So that is not an issue, but looks like the bound state is not doing very well. So what we learned was that the simple geometrical structures of just solo RNAs, such as microRNAs and others, can be more useful in predicting sequences of a riboswitch that is not seen in the training data set. Here are TPP riboswitch. This is even more so than using other riboswitches. So in other words, what we learned was that the geometry we take, the geometrical information we learn from other riboswitches may not be as useful in finding a novel riboswitch, which is not great. That means known riboswitches don't give us a lot of information about a novel riboswitch. Every riboswitch is very different. And we've seen that in a different way in our results here. We also learned that unbound structures are easier to predict, but the bound structures have more complication, which we're currently working on. So in our next step, we are interested in experimental validation of some of our prediction that are very close, as well as improving our models by applying different mathematical techniques or different kind of combinatorial techniques to combine information, as well as considering other biological factors such as metabolites. They affect structures. As you can see here, the G quadruplexes can form differently when there's metal ions around, which was not even considered in our model. So our model has a long way to go, and this was fairly simplistic in terms of just looking at the geometry of base pairings, but we are looking to build on that. And I would like to thank the other authors and collaborators from Johns Hopkins and Roma universities, and I'm very grateful for their input and collaborations in this work. I also thank you for your attention and looking forward to answering any questions online or via email. Thank you very much.